Carlos, I hope you will tell the participants in brief what you will speak about today, and then afterwards, Anders will introduce you.
Well, thank you for first of all, thank you for the invitation. I'm super happy to be involved in this type of activity, and hopefully everyone will enjoy the webinar. So I'm going to discuss, my main background is in biomarker discovery. We have been working, I have been working in extracellular vesicle research for the last probably 8 years. So what I'm going to discuss during the seminar is the role of extracellular vesicle, mostly coming from the placenta in diabetes pregnancies. So hopefully everyone will enjoy the seminar.
Thank you, Carlos. Anders Askeland, will you please introduce Carlos Salomon?
Yes, sure. Thank you, Hannah, for the introduction, and of course, thank you to all the participants for being here today. Firstly, as Hannah already said, my name is Anders, and I'm currently a PhD student, and I also work within the field of obesity and these extracellular vesicles, which of course, very coincidentally, is somewhat related to what Carlos is going to speak about today. And of course, with that, it is my pleasure to introduce and welcome the speaker for this webinar, Carlos Salomon. For those of you who don't know Carlos is an associate professor at the University of Queensland, Australia, where he has both established and now leads his own independent research groups called the Exosome Biology Laboratory. He is also an internationally recognized researcher within the field of extracellular vesicles and pregnancy, where his research focuses on these vesicles in relation to pregnancy, preeclampsia, ovarian cancer, but also in relation to using EVs as clinical biomarkers. Some of his main contributions to the fields include having increased the understanding of the role of placental exosome during gestational diabetes, among other things. Additionally, Carlos is working on the identification and validation of plasma exosomal biomarkers for complications during pregnancy, and while doing all of this, he's also trying to establish and develop EV biomarker discovery tools using transcriptomics and proteomics. During the course of his career, he has won several awards and been appointed to several positions, and also been awarded many grants. One of these latest grants being the Danish Diabetes Academy Visiting Professorship grant, where Carlos has received funding to visit, among other things, my group at Aalborg University Hospital, but also other groups in Denmark, where we will, in collaboration, investigate stuff like extracellular vesicle cell-to-cell communication and obesity, biomarker discovery for the metabolic syndrome and diabetes, and characterization of EVs during pregnancy, something we are all very excited about. Now, without further delay, here is Carlos and his presentation.
Thank you, Anders. Carlos, please start your presentation. And your PowerPoint looks perfect.
Okay, fantastic. Thank you. Well, thank you for the very kind introduction. I'm super happy to speak today and trying to share what my group have been doing for the last 7 to 8 years. So I know that the time is a little bit limited. So today I'm going to talk mostly about extracellular vesicles and how these vesicles can communicate, coming from the placenta, can communicate with other tissues in the context of gestational diabetes, right? So in this first slide, you can see this is the title of my presentation, but also my email address is over there, and my Twitter account, and also the lab Twitter account is there in case that you want to establish a collaboration or at least have a further conversation or discussion about the presentation. More than happy to have that type of discussion. Okay, so. Okay, so I don't have anything to discuss. And in the picture here, you can see this is the picture of the UQ Centre for Clinical Research, which is located in Brisbane, Australia, and my lab is there. It's a first-class facility in which we have been developing different methods to isolate and enrich different types of extracellular vesicles in the context of pregnancy, but we have also the project running in different diseases as well, right? This is our program. So we focus in extracellular vesicle. And you can see here is essentially is kind of a pipeline in terms of how we run our research. So we focus in isolation or enrichment of different types of extracellular vesicles, which mostly are known as an exosome for the small ones. And this can be isolated from different biological fluid or also from cell conditioned media. So what we do with this vesicle, we characterize them. So we are able to characterize, for example, vesicles from different origin when we isolate or enrich vesicles from biological fluid like plasma. Also, we provide the content of this vesicle, which can be used as potential biomarker discovery using mostly mass spectrometry approach. We have great facilities in the center, so we do protein identification, but also protein quantification using mass spec. Also, it's well known that extracellular vesicle can also carry different type of molecules, including microRNA. So we focus a lot on microRNA, and this is part of the data that I'm going to show later on. And then, of course, when these vesicles are released from different cell types, they are interacting with other cells, right? So we also have different experiments in which we can evaluate what is the target of this vesicle, what is the role, and potentially identify therapeutic targets associated with the specific diseases, right? So this is like the context of my lab, and that is what we do, right? So for the presentation, I want to just mention just three main components of the presentation. So of course, it's going to be about extracellular vesicles, and mostly it's going to be I'm going to talk about vesicle coming from the placenta here in the picture, and how this vesicle in the context of high glucose, insulin, and also diabetes condition can interact with mostly insulin target organs like skeletal muscle. But also we have been exploring the possibility of adipose tissue, how the adipose tissue secrete extracellular vesicle, and that can interact with the human placenta during pregnancy. Just to start, this is a very fancy but very simple but very good video about why extracellular vesicle is kind of a hot topic in the literature at the moment. So you can see this vesicle can carry bioactive molecules like protein and RNA. They're involved in cell-to-cell communication under normal condition and also under pathological condition. So that means that when one cell releases this extracellular vesicle, this vesicle can interact with other cells and release the content, right? And that content will modify the biological function of the target cells. It's a lot of information about how vesicle can regulate immune response, intracellular communication, metastasis, angiogenesis, and other as well. In terms of the type of vesicle, so this is a cartoon about biogenesis of extracellular vesicle, in which we can divide mostly in two main type of vesicles. So vesicle mostly coming from the plasma membrane and vesicle coming from an endocytic origin. So this is the vesicle that usually we call exosome, but still it's ongoing discussion in the literature about what is probably not the correct name, but the more informative name for extracellular vesicle, right? So if you divide that, you have these extracellular vesicles that are you can see in the early endosome, and then they form the intraluminal vesicle, which can be transferred to the plasma membrane and then released as a exosome, right, or a small EVs. Also, you have vesicle which seem to be bigger as well, coming mostly from the plasma membrane, and it's very interesting because I think, and it's a lot of data in the literature about that. Mostly, I think the main difference between the different type of EVs is about the biogenesis, and that potentially can also influence the content of this vesicle and therefore the function of this vesicle. Then these vesicles are released, and then they can interact with the plasma membrane with the target cells and release the content. It's several gaps in the EV's research, and probably one of the biggest gaps is about targeting, right? So are these vesicle coming from a specific cell type already predetermined to interact with a specific target, right? Which is, we don't know. It's a lot of hypotheses about a potential barcode in the membrane of the vesicle that you can track and then, for example, determine where this vesicle might go, right? But in terms of the diversity or heterogeneity of extracellular vesicle, also is more complicated just than exosome or microvesicle or microvesicle. And this is a very nice publication in which you can see that everything seems to be more complicated, right? And when you separate the different type of vesicle by size, you can see that you have even the small one that are the exosome size, you can divide in three main categories as well. So you have the exomer, the small exo, and the large exosome. And then you have other different type of vesicle which are bigger as well, right? And why this is quite interesting, because also is information in the literature that this different type of vesicle also have different function, right? And that is true, they are different in the content as well, right? If you don't like the cartoon, this is a electron microscopy image in which you can see the multivesicular bodies here in the cell. Inside of that are the intraluminar vesicle, which then is going to be the extracellular vesicle released from the cells. So how we can study extracellular vesicle? We use two main approach in the lab. So we can use cells. So you culture cells from normal or pathological condition. You can use cell lines and culturing this cell under different microenvironments to try to mimic the normal and physiological condition. And then you get the vesicle from the cell condition media. You can provide the content of this vesicle. You can also determine the level of this vesicle. For example, if high glucose, insulin, and others factors associated or molecules associated with diabetes can regulate the release of this vesicle, right? Also, you can isolate it mostly from all biofluids, more common from plasma or serum. And then using a similar approaches, you get the vesicle, and then you have your experimental setting with your controls and then your different disease conditions. You can determine if they are any different in the concentration of the vesicle or maybe in the content of the vesicle. Usually in my lab, we run proteomic discovery projects and also microRNA analysis to determine if we can identify specific protein or microRNAs associated with different diseases, right, as a potential biomarker discovery. One of the big questions here is, okay, how you isolate or how you enrich extracellular vesicle, right? And it's a very good question, and it's hard to answer. I think it's not a correct answer. It depends what is the question that you're trying to answer, right? So you can, this is also a very nice cartoon in which you can see separate the method to enrich EVs as a concentration or purification method. It depends what you're trying to answer, right? It's very interesting that essentially the concentration method seems to be more yield, so you get more vesicle, but also you get a lot of non-vesicle material that might be a problem when you're trying to identify potential mechanism and effect of this vesicle. From biomarker discovery, potentially is not a problem as long the method have a good reproducibility. And then you have other method in which you have a more pure population of vesicle, decrease the non-vesicle material, for example, side-clutching chromatography, which is very popular at the moment because it's less time consuming compared to, for example, a density separation, which have been very popular in the past as well, right? So again, I think probably I can be talking about method for another 3 hours, which is not the real aim of the presentation. So probably if you're interested in extracellular vesicle and your first question or your first decision is how you're gonna get the vesicle, it depends on what you're trying to answer, right? What is your research question? And the other thing is trying to be consistent as well, trying to characterize what you get, right? So trying to use different methods, characterize the vesicle by proteins. For example, what are the proteins of the vesicle that you're isolating? Trying to characterize also the non-vesicle material to check how much you get there. Morphology, electron microscopy, to trying to see the vesicle, and of course, this different method to provide the size of this vesicle, right? So in this context, we have been focused in identification of potential biomarker for gestational diabetes and focus in cell-to-cell communication during pregnancy. So I just would like to point out three main concepts about gestational diabetes. It's one of the more, the incidence of the gestational diabetes varies according to the country, but in some of the countries it is even 20% of the pregnancy, right? And it's a big problem because it's associated with fetal overgrowth, increase of the perinatal morbidity and mortality, but also increase the susceptibility of these women to develop cardiovascular problems and also for the babies later in life. Also, women that have gestational diabetes, they have a higher risk to develop diabetes type 2 later on. And currently, there is no effective treatment. So you have insulin, diet, and other pharmaceutical therapies that you can use, but they are not very effective for the long-term effect of gestational diabetes, right? And mostly because the pathology is not fully understood as well, right? So in this context, because it's pregnancy, we have been focused in the human placenta. So you are not familiar with the human placenta. Here's a real picture of the placenta. You have a fetal side, which is connected to the baby here, and then you have the maternal side here, right? So if there are many different amazing things about the placenta, don't worry about number 7. But probably one of the more interesting things about placenta is that they separate the maternal versus the fetal circulation, right? And you can see in this cartoon, we're interested in the chorionic villi and specific in the layer of the syncytial trophoblast, because the syncytial trophoblast is in direct contact with the maternal circulation. So we are trying to understand how the placental cell can, for example, sense the maternal microenvironment, and in response of this maternal microenvironment, for example, diabetes microenvironment, can increase, decrease, or change the release of this extracellular vesicle, and also modify their content, right? And this is the cell that we are culturing because they are in direct contact with the maternal circulation. So this is a study that we published several years ago, one of our first studies, in which what we did was isolate trophoblast cells, and then we wait for 72 hours to obtain mostly syncytial trophoblasts, right, which is in direct contact with the maternal circulation. And what we did was expose these cells to different oxygen tension, right? So hypoxia is associated with different complications of pregnancy and also different diseases, right? To identify if hypoxia can induce changes in the release and content of this extracellular vesicle. And you can see that hypoxia on these cells can increase the level of extracellular vesicle, right? And then when we did this was at least 7 years ago. So we performed a proteomic analysis, and we identify a specific protein associated with hypoxia in this extracellular vesicle. Then we combine hypoxia and glucose concentration. So we expose the cell to low oxygen tension and then also high glucose, trying to mimic what is happening in the diabetic environment, to identify if you can see a similar difference. And you can see that this effect of hypoxia is even higher when these cells are exposed to high glucose as well. So what this suggests is, if this happening early during gestation, right, perhaps it's a signature that we can track in the biological fluid, in the plasma of these women, that we can identify women at risk to develop complications of pregnancy later on. And this is the hypothesis that we have been working for several years, in which if we can profile, for example, the placental vesicles in early during gestation, perhaps we can classify women at risk to develop different complications of pregnancy, including gestational diabetes, right? So to test that hypothesis, what we did is isolate EVs from plasma, right, or serum. In this case, we use plasma. It seems to be that it's more vesicle coming from platelets in serum, so that's the reason we start our program with plasma, and we're trying to be consistent, use plasma mostly of the time. I think we have been using serum just a couple of times when plasma is not available. So the first question, when we moved to work with extracellular vesicle in plasma, our first question was, well, can we use plasma that have been already stored, right? Which is probably a very big question, because essentially get blood, isolate plasma, and isolate the EVs right away is not feasible when you are trying to perform a larger project, right? So this is the first thing that we did, and we isolate EVs for fresh plasma and plasma that have been stored for this particular paper 3 months, because have to be from the same patient, right? And we didn't identify major difference in terms of how many vesicles we get. The characterization of the vesicle were very similar, and also we provide some content of this vesicle in terms of the protein and microRNA. There are not many difference. So we think that they are very stable, and therefore you can use samples that have been already stored in different biobanks to run your research as well. So then what we did was quantify vesicle in GDM pregnancies. So you can see this is a paper that we published several years ago, in which we quantify circulating vesicle, placental vesicles in GDM pregnancy. So in this figure, you can see that in general, in GDM pregnancy, the level of circulating placental vesicles are higher compared to normal pregnancy here. And then when you classify this according to the gestational age, you can see that you have a consistent increase very early during gestation. As the GDM is diagnosed in the second trimester, we thought that how good could be the analysis of total exosome and placental exosome as a potential classification model to identify women at risk to develop gestational diabetes. So we developed this, and you can see that if you quantify, at least in this cohort, if we quantify the number of placental vesicles, you can see that the positive and negative predictive values of use these placental vesicles in GDM pregnancies is quite fantastic, with almost 90%, right? So suggesting that this can be a potential good biomarker to classify women at risk to develop GDM later on, right? But also we're interested in the function of this vesicle, and of course you're talking about gestational diabetes, which is associated with insulin resistance. You need to talk about how the skeletal muscle, for example, can sense insulin, and in response to insulin, increase the glucose uptake, right? So we're trying to determine if there are any effect on the placental vesicle on the skeletal muscle, right? So for that, what we did was isolate EVs from chorionic villi for first trimester pregnancy, and then we provide a macroRNA sequencing to determine if there are any difference in the content of this vesicle coming from normal and GDM pregnancy. Sorry, these villi were isolated from for pregnancy of term, okay, not for trimester, because we need to know if they develop or not gestational diabetes. And then we identify, you can see this is the macroRNA analysis for chorionic villi macroRNA analysis in normal versus GDM, and we identify a different set of macroRNA that are up and down regulator in GDM compared to normal pregnancies. And then we did exactly the same, but in the vesicle coming from this chorionic villi explant, and you can see that we identify also a set of macroRNA, and we identify a set of macroRNA that the difference in the villi is reflected in their derived exosome, right? So we decide to check if this vesicle, which these macroRNA, which are different in this vesicle, have a pattern in terms of what is the expression in the placental cells, in the exosome, in the circulating exosome during pregnancy, but also in the skeletal muscle, trying to identify a potential delivery of this macroRNA into the skeletal muscle. So you can see that we identify, there you go, we identify a pattern of this macroRNA expression in the placenta, in the circulating exosome, and the skeletal muscle, right? Was a similar pattern, suggesting that maybe this vesicle can be released from the placenta and deliver this macroRNA into the skeletal muscle. Then what we're trying to identify what is the potential effect of this extracellular vesicle. So on a skeletal muscle, for that we use two different skeletal muscle cell lines, primary cell coming from normal and diabetic patient. And then it was very interesting. And then what we did was determine how this vesicle, how the skeletal muscle respond to insulin in terms of the glucose uptake. So you can see, probably when I just jump to the main results. So you can see here that when incubate the skeletal muscle with exosome coming from normal pregnancy, you can see that the normal skeletal muscle is still sensing insulin and essentially no effect. But when we incubate the skeletal muscle with exosome coming from GDM pregnancy, you can see that the effect of insulin was completely abolished. So essentially, the glucose were unable to increase the glucose uptake in response to insulin, which is essentially a definition of insulin resistance. So suggesting that the potentially one of the role of this vesicle could be changed insulin sensitivity through delivering of molecules into the skeletal muscle. So then we're trying to expand this research and trying to identify macroRNA in a larger cohort of sample. So what we jump on was trying to identify macroRNA in the circulating vesicle across gestation, to identify if we can identify macroRNA associated changes in GDM pregnancy across gestation, right? So for that, we use a discovery and validation cohort patient, a cross-sectional for the discovery, and longitudinal set of patients for the validation. So this is just the experimental design. We isolate the vesicle, we characterize them, which is important analysis that you have to do if you isolate EVs, protein associated with EVs, site distribution, and also identify the percentage of macroRNA associated with EVs, at least in our preparation, right? So then what we did was as a macroRNA sequencing, and when you do sequencing, probably the first and big decision that you have is how you're going to analyze the data. So we decide to perform a linear modeling analysis to identify macroRNA that change across gestation in normal versus GDM. We identify around 300 macroRNA that change across gestation in normal versus GDM in the circulating exosome, and then we select 9 macroRNA that were fully validated in the validation cohort. So you can see this is of the 9 macroRNA, and 7 of the 9 were validated in an external cohort patient, suggesting that GDM is associated with changes in the macroRNA content across gestation. Then we develop a classification model using this macroRNA, check if we can provide this macroRNA early in gestation, we can identify women at risk to develop GDM. And you can see that when we perform that in the individual macroRNA, we didn't get good results, but then when we develop a classification model using the combination of all these macroRNA, you can see that we are 100% positive and negative predictive values to identify women at risk to develop GDM later during pregnancy. It's very interesting. Of course, I mentioned about the skeletal muscle. So also what we did is get skeletal muscle biopsies for women with gestational diabetes at the moment of delivery, identify potential target of this macroRNA. And you can see that the set of macroRNA that we identified previously were associated with protein and genes, which are associated with insulin signaling and glucose homeostasis. And then we identify a pair of macroRNA and genes and protein that are associated with mostly STAT3 signaling in this context, suggesting that circulating macroRNA associated with exosome or extracellular vesicle can potentially be targeting this protein and genes in the skeletal muscle in the GDM. So what we're proposing here is, at the moment, essentially just a little bit change the dogma and suggesting that also are the extracellular vesicle involving changes in systemic glucose homeostasis during gestation. However, all this data that I show is mostly in vitro data about the mechanism, but also plus the analysis from plasma. So when you move from the in vitro to the in vivo analysis, not always works, right? So we're trying to test this hypothesis. What we did is inject a vesicle from diabetic pregnancy into mice and check if we can change the glucose systemic homeostasis in vivo, right? And this is a collaboration that we did with Thomas Johnson at the University of Colorado in Denver, and we published the data. This is I'm going to show you just the main result, but this is the full article in case that you want to take a look at the article. And one of the good thing that we did in this particular article is rather than to inject vesicle in mice, Thomas developed an osmotic pump to infuse the vesicle in mice. So essentially, it's a continuous release of vesicle for 4 days in these mice, which seem to be mimic much better the physiological condition, right? And this is probably the main result. So you can see that how the vesicle coming from GDM pregnancy can induce high blood glucose level in these mice and increase decrease insulin sensitivity in these mice. Again, supporting and give now in vivo evidence about this hypothesis about the exosome, and mostly the placental exosome, can induce glucose systemic changes in glucose systemic homeostasis during gestation. The other thing, very interesting thing that we did in this publication as well, that Fred did, was isolate the beta cell from the pancreas from these mice, and then check if the exosome and also regulating the secretion of insulin. Amazingly, you can see that how the GDM exosome can actually abolish this secretion of insulin that you see in the presence of normal exosome from normal pregnancy, right? Now, adding to this hypothesis that not only this effect could be through the skeletal muscle, but potentially can be interacting with the pancreas and with the beta cell as well, right? So what we're thinking is that the placental exosome can maybe interacting with insulin target organs and regulate glucose systemic homeostasis during pregnancy. I'm almost done with the time, so I'm gonna be very quick in the next 2 minutes. The final story that I want to tell you is about adipose tissue, which is very interesting as well, right? And why is that? Because of course, BMI and obese women have a high risk to develop GDM during pregnancy, right? And this is a very nice study that we published a couple of years ago, in which we identify a correlation between BMI, which is here, and the level of vesicle during gestation, right? So then suggesting that potentially not only the placental vesicle are involved in this phenomenon, maybe it's a signal coming from the adipose tissue that potentially we can identify. So for that, we did a deep proteomic analysis in circulating EVs in GDM pregnancies, and from normal and GDM, you can see here the different proteins were identified. But what we did was isolate total EVs, circulating EVs in this pregnancy. So the big question is, from where are these EVs coming from, right? So we did a bioinformatic analysis, and we identified actually in GDM, you have an enrichment of circulating vesicle coming from the placenta, which is consistent with what we have been also demonstrating with different publications. But surprisingly, we identify enrichment of adipocyte signal, suggesting that maybe the adipose tissue also is contributing to this signature that we see in circulating exosome, right? So what we did was trying to develop this a little bit further, and Nantini, what she did was culture adipose tissue explant and then isolate the EVs from this culture, and then you can see the explant coming from GDM actually have higher level of exosome, and then it's a linear correlation between the Z score, which is suggesting that potentially this vesicle can be involved in fetal growth, right? Then we did a in vitro analysis. We get exosome from adipose tissue, we incubate on placental cell, and we check glucose metabolism, right? And then you can see here that we identify different genes associated with glucose metabolism that respond to this exosome coming from the adipose tissue from GDM women, right? So to simplify the analysis, we identify that actually adipose tissue GDM exosome, you have a higher signature associated with glycolysis. We're suggesting that potentially there can be an effect in fetal growth, right? So finally, what we did was to test this hypothesis. We injected adipose tissue exosome from normal and GDM in pregnant mice and determine if we can see any effect on fetal weight and placental weight. So you can see how the adipose tissue vesicle coming from GDM, you can see a higher level of placental weight and also a higher level of fetal weight, suggesting that they are involved in fetal growth, right? So this is the main summary of this. We suggest that maybe it's a cross-link between the placenta talking with insulin target organs, but also adipose tissue maybe is also talking with the placenta during pregnancy. So finally, we know that the placenta and also the adipose tissue release small EVs like exosomes, and they have different distinct molecular and biological properties. Also, we identify specific macroRNA changes associated with GDM, and also exosomes from GDM placenta decrease insulin, stimulate glucose uptake in skeletal muscle, and seem to be that the adipose tissue is interacting with the placenta and regulate fetal growth in GDM. Probably the more important slide, this is just acknowledgement to my group. I'm a very lucky researcher. I work with a very smart people, and I can do anything without them. So thank you very much. Also have fantastic collaborators in Australia, overseas, in Chile. We have plenty of different collaboration associated with pregnancy as well. And of course, my collaborators in Denmark, I had the opportunity to went to Denmark before the pandemic 2 years ago. I had a fantastic workshop over there. I'm now looking forward to be able to travel again soon. Thank you. Happy to answer questions and discuss this a little bit further. Thank you.
So I guess it's me now. Thank you for a great presentation, Carlos. So there's already some questions. I guess other people also can write, but I can start with a couple of my own very quick ones that I kind of got. And the first one being, how do you talk about placental vesicles or EVs? How do you define these and know that they are in fact placental vesicles?
Yeah, so that's a very good question, because when you get, for example, when you culture placental cells and then you get vesicles from the cell conditioned medium, they have to be placental vesicle because you have only one cell type in vitro, right? Then when you move to biological fluid, you get a total vesicle isolation or total exosome, right? So it's coming from everywhere. So for researchers working in pregnancy, we are lucky to have a specific placental marker with the placenta, the placental alkaline phosphatase PLAP. So we can actually select the EVs coming from the placenta using this marker, right? From plasma.
Okay. Then a quick follow-up. How do you select based on these markers? You are doing proteomics and transcriptomics, among other things.
No,
do you affinity concentrate them?
You know, what we do is if you want to select placental vesicles which are circulating during pregnancy, we do a total EVs isolation from plasma, and then we do an enrichment using immunoprecipitation using PLAP.
Okay, thank you. And then I can take some from the participants, I guess, attendees. I apologize if I kind of butcher the names here, but the first one is from Atonia Hufnagel. I believe there's two questions in here, so I take one and one for you. First one is being...
Anna, I think she wrote a comment below that Carlos had already answered her first question. She posted She posted the question before he finished his presentation.
So this is only the second question.
Yes,
then.
I think.
She's asking, in the experiment infusing human EVs into mice, do you think the species difference affect EV uptake or tissue specificity? For example, does PLAP not being present in mouse?
Yeah, look, this is a fantastic question, right? Because when we get exosome from plasma and then we infuse in mice, our first concern was that, well, potentially this can induce a pro-inflammatory state, and this pro-inflammatory state, because it's human to mice, can change the glucose system homeostasis, right? So I have two answers for that. The first one is we did experiment. So what we did was check what is the effect of human exosome in activation of T cells in the presence of this human exosome, and we didn't find any. Also, we provide pro-inflammatory markers in the mice, and we didn't see much difference in the presence compared to the absence of the exosome, right? But look, this is a fantastic question, and also PLAP also is expressed in mice as well. So yeah. So look, it's not a perfect marker, but I think it's good enough to enrich the vesicle. Yeah. But the potential immune response of injecting human vesicle in mice is a very good question, and we did experiment to prove that at least the change in systemic glucose homeostasis is not due to the potential immune reaction of this human exosome.
Great, thank you. And then the next one, I believe, is from Åsa Handberg, which is asking, which approach do you take to identify new cell-specific surface markers of EVs? For example, those to be used for immunocapture.
Yeah, well, I haven't been able to identify new markers, right? Which is something that we try, to be honest, because probably the approach that we're trying to use is trying to isolate membrane protein from, you have this kit in which you can have an enrichment of membrane proteins. Then you can profile, identify what are the markers in the membrane of the vesicle or of the cells, which can be associated or can be specific, right? However, we have an issue about how much we need to identify in vesicle, because these kits that are designed to get membrane proteins from cells are not designed from EVs, and of course the amount of material that you have is much less. So we didn't have much material when we're trying to only isolate the membrane protein in the vesicle and then potentially identify a potential specific protein. The other approach is what we do here is in my lab is global proteomic analysis. So when we get everything from the vesicle, then you can separate the protein that you identify according to the location, and maybe you can select the protein, the membrane protein there. But again, we haven't followed up that particular idea much. And the main reason is because we already have a good marker for placenta, which is the plaque, right? For my other project in cancer is something that potentially we want to pursue later, but it's a big question. And if we have able to identify a specific marker associated with vesicle, will be a tremendous advantage, because then you're going to be able to isolate a specific type of vesicle from plasma or serum, right?
Thank you. And then the next one is from Rosa Jurlund. Well, which only says, thank you for a very interesting presentation, I guess. But the next one is from Jürgen Clems, which asks, do you take any precaution about diet before taking plasma sample? He also has one more, but I can take it afterwards.
Yeah, well, so for our GDN cohort, we have information, for example, which women get, they have a diet or insulin treatment to treat the gestational diabetes. But our group are quite mixed, so we don't have much information about. What we have is BMI, which is one of the clinical parameters that we take care, and trying to, when we separate the group, trying to have a similar BMI across the group, so we are not influenced by the BMI. But again, I think it's hard to control unless you design a specific experiment to identify what is the effect of diet on the circulating vesicle, right? But at least for the cohort that we have available, the best information that we have is what women have been treated only with diet versus insulin during pregnancy, and the other is the maternal BMI, which also we take in consideration all the time.
Great. So Jürgen also have two more questions. Do you find bacterial sequences from EVs? And if yes, how much do you find? He asks.
If I found what?
Bacterial sequences, I guess, in the mRNA or the transcriptomics.
Yeah. No, I haven't seen any, at least. It's a different. I don't work with EVs coming from the bacteria, but it's several groups working in that area. But I don't have much expertise on that particular topic. Yeah.
Yes, great. And then there's another one from Orsa, which asks, have you taken the other way around approach where you would identify EV populations that exert specific effects on a target in order to characterize a specific exosome populations?
So when you say, I try to understand what you're trying to answer, but I guess the other approach is trying to focus in the vesicle, focus in the target, and check if you can see, we can see if there is any specific vesicle interacting with this particular target. If she mean that. No, no. Usually, what we do is isolate total vesicle from plasma or from serum, from urine, from saliva, and then what we do is through our proteomic analysis identify from where this vesicle coming from. But then it's very hard to isolate a specific subpopulation of vesicle from biological fluid because you don't have a specific marker. We're lucky that we have blood from the pregnancy story. But it's hard. Also, the yield probably is not going to be good enough. If you're interested in, for example, endothelial cell-derived vesicle, potentially you can use CD31 to enrich vesicle coming from the endothelial cells. You can use a platelet marker if you're interested in platelet exosome, right? Yeah.
Great. Just the last one is, I believe, somewhat answered, but I will just ask it anyway.
Yeah.
It's from Victor Galatia, which asks, have you taken a look at liver tissue exosomes in your studies?
No, I haven't. I would love to, right? Look, one of the I would love to do everything, right? But no, we haven't. But look, I think the EV research have been growing exponentially, right, for the last 5, 7, 10 years. So I'm pretty sure that someone is focused only potentially in liver EVs, right? And what could be the effect in cell metabolism, for example. But we haven't. Even this paper that we have about the effect in the pancreas and the beta cells is something that we did through collaboration because it's not something that I do. So if someone is interested in liver EVs, more than happy to have a chat.
Great. I believe
we will have time for 1 or 2 more questions, but you will have to write them in the chat now. And meanwhile, I will open a poll, which I hope you will all take your time to fill in. And remember, if you have a question or comment, you can also ask your question verbally. Just write your name in the chat, and then we will unmute you.
That was me. Thank you for the question.
Yes.
Yeah, so look, I think just to try to summarize, we have one more minute. If you're interested in extracellular vesicles, of course, in the diabetic context, I think that there are 3 main areas that people have been exploring, and I'm doing that in my lab as well. One is regulation of the secretion of this vesicle, right? So how the environment how cells can send the different environment and release different population of EVs. The second part is about the content. So if, again, how the cell can incorporate a specific protein and microRNA in extracellular vesicle. We have a lot of data, and it's something I didn't talk about it, but we have a lot of data about a specific protein that are involved in the packaging of microRNA in the exosome, which are not necessarily enriched in the originating cell. So you have a specific package of information in EVs. And the third part is about interaction with the target cell, right? This barcode, how you can identify a barcode in the EVs so you can use a specific type of EVs for delivery, right? Delivery therapeutic agent, etc., right? So I think this is the main 3 areas that if you are interested in EVs, you can explore.
Yes, thank you, Carlos Salomon, for this summary. And as there seems not to appear any more questions in the chat, Anders, I assume you do not have any more questions in your feed. Then
No,
I think, great. Then it's time to wrap up. I want to thank you all for attending the webinar, and then most of all, I of course want to thank Carlos Salomon for presenting his research at this webinar, and Anders Eskeland for being chair at the webinar. I wish you all a very nice weekend. Bye-bye.
Thank you.
bye.